So I'm going to be talking only partially about SARS coronavirus 2. I thought the topic that I was given was recent efforts in viral vaccines, and obviously this is a very large topic. So I'm going to try and address just components of that. This is the outline of the presentation, an introduction, the India rotavirus story. I'll talk about Ebola, challenges in making vaccines for pandemics and epidemics, and they cover SARS-CoV-2 and next generation influenza vaccines, and end with that. So coming first to thinking about vaccines, vaccines have been with us only for a little over 200 years. In the 1700s, in Europe alone, 400,000 people died every year of smallpox, and it was Edward Jenner and Benjamin Jesty's experiments in the late 1970s that showed us that if you had an inoculation with cowpox, you could protect against smallpox. The demonstration of these infections, what is shown here is the very famous illustration of Edward Jenner with James Phipps. This was actually the first human challenge study that was documented, because James Phipps was given cowpox and then challenged 2 months later with smallpox. Now, we were able to eradicate smallpox almost 200 years after the vaccine was developed, but it's very important to understand what vaccines can do to disease. Shown on this graph is the example of measles. You can see that since documentation started, we had in the US, outbreaks of measles that continued for a documented period of 20 years. And then as soon as the vaccine was licensed and introduced, the cases of measles just crashed. When a disease goes away, people seem to think that the vaccine does not really matter. And we have had recently many outbreaks of measles in the United States, resulting now in many places asking for mandatory immunization with measles vaccines. So if we look at what vaccines do, it's estimated that vaccines prevent 2 to 3 million deaths a year, 7,000 deaths a day, 300 deaths an hour, and 5 deaths every minute. And this is counting only deaths, not counting all the sickness that can be prevented by vaccination. If we look at vaccines and the making of vaccines, which is what we are interested in at the moment. This is counting only, that's not. Sorry. If we look at making vaccines, it can take decades. If we look at methods used to make vaccines up until the 1950s, we followed essentially the same principles that Jenner and Pasteur had used, which is let's isolate the organism that causes the disease, inactivate it or attenuate it. In Jenner's case, find an attenuated version by taking a strain from animals, inject that into human beings, and then wait for an immune response that is protective but does not cause disease. So we wound up with bacterial vaccines that are responsible for much of the childhood immunization that we have today, as well as a range of viral vaccines that were made using these methods. After 1950, we've had many new technologies. So cell culture, for example, allowed us to make vaccines against polio, hepatitis, mumps, rubella, and the rotavirus vaccines that we make in India today. In 1980, we had recombinant DNA technologies. These allowed us to make acellular pertussis vaccines, hepatitis B vaccine based on the surface antigen. In 1990, we got glycoconjugation, which allowed us to make vaccines so that we could have a durable immune response against what were initially T-independent antigens. In 2010, we had reverse vaccinology, where you could take the sequence of an agent, identify immunogenic epitopes, then make those and test them out in animal models and subsequently in humans. The first vaccine that we had licensed using this strategy was a meningococcal vaccine, and there are many other vaccines that are in development using these approaches. But really, it is during the last 20 years that we've had many, many new technologies, and many of them have built on what we know about viral vaccines, particularly focusing on the so far unsuccessful attempts to develop an HIV vaccine. These include structural vaccinology. They include new attenuation methods, such as the codon de-optimization techniques we hear about, vectored vaccines, synthetic biology, the ability to make proteins, RNA, and DNA vaccines. Our understanding of adjuvants and what we can do with them grows every day. And more recently, because we've been able to do single cell separation of plasmablasts, we've been able to better dissect the human immune response in order to identify antigens that may make suitable vaccines. So moving from all this exciting new technology, let me tell you about how hard it really is to make a vaccine by walking you through the rotavirus story in India. So if we look at rotavirus, it is viral gastroenteritis, the commonest cause of severe dehydrating gastroenteritis in young children. It infects children who are rich, children who are poor. It spreads easily, and improvements in hygiene, sanitation, and drinking water don't adequately prevent rotavirus infections. So if we look at the virus, it is a virus about which a lot was known, that was adapted to cell culture in the late 1970s. And almost as soon as the virus started to be grown, people started looking at making vaccines. There were a lot of efforts that I'm not going to go into, but the need for the vaccine was obvious very, very early on. Even in the 2000s, we found that if we looked at diarrheal deaths around the world, 37% of them were due to rotavirus. In India, it's estimated that before we introduced vaccine, we had at least 60,000 deaths a year. The earlier estimates were over 200,000 deaths a year. We also found through many of the studies that we had done that this had not just a significant health burden, it also had a significant economic burden, because one child's hospitalization resulted in an expenditure that was equivalent to 5% of annual household income. Now, if we go back to how long it took to make an Indian vaccine candidate, the story starts and to some extent ends with Dr. M.K. Bhan, who was a young pediatrician at AIIMS, who identified at a time when other people who are still in and around Delhi showed that there were rotavirus infections in the neonatal nursery, and then not leaving it at that finding, followed up children who had had neonatal rotavirus infections and demonstrated that those who were infected in the nursery had fewer attacks of rotavirus diarrhea than children who had not been infected in the nursery. They then worked with the Indo-US Vaccine Action Program to make and grow the virus, then brought the vaccine back to India, licensed it to Bharat Biotech in Hyderabad, and worked with ICMR to continue to define the burden of rotavirus disease while in parallel developing the rotavirus vaccine through studies that were done at the Society for Applied Studies, where much of the testing was done at THSTI. If we look at the phase 3 study of the rotavirus vaccine, the vaccine itself, the strain was identified in the 1980s. It was developed in the US in the 1990s. It was brought back to India in the early 2000s. And the phase 3 efficacy trial was done in the around 2008 to 2013. It took about 5 years. There were 6,800 children, and there were over 300 people who made up the study team. The vaccine was evaluated in Delhi, Pune, and Vellore. But in Vellore, we had already been doing studies that demonstrated that the vaccine would not work as well as the vaccines that had been developed outside the country. We predicted, based on the data that we were generating, that the vaccine would have approximately 45 to 50% efficacy based on data that we had generated from our cohorts. But even with that, we thought if we have a 50% efficacious vaccine, that would prevent about 30,000 deaths in India every year. When we got the results of the trial, we showed that we were not too badly off. The vaccine was preventing 55% of severe and hospitalized rotavirus gastroenteritis. Once we had the vaccine available, the decision had to be made by the government of India whether the vaccine should be introduced. And that, of course, depends on the disease. We had shown that there was ample disease burden. We had shown that the vaccine worked. And the government thought that the immunization program and health system was strong enough to accept the vaccine. So the vaccine was, we had done the cost effectiveness studies, and in 2016, the vaccine was introduced. Initially for 9% of the birth cohort, and in 2018, another new rotavirus vaccine was introduced. In 2019, rotavirus vaccination was scaled up to the whole of India. We had been looking to see whether this vaccine would work in the real world or not. And we have now generated data that if you look at children, you do see an approximately 50% efficacy of the vaccine, but the vaccine doesn't work well in all children. If you have stunted children who are in the second year of life, the vaccine does not work at all. So even though we had a rotavirus vaccine, which, as I said, was based on technology developed from the 1950s, there is a clear need for new rotavirus vaccines. And the goals here are better performance of vaccines, better than the 50% we see now, greater safety of the vaccines, because rotavirus vaccines are associated with intussusception, and we need vaccines that are cheaper, and we need the potential for combination vaccines so that we can reach more children with more antigens easily. So now there is work that is ongoing on non-replicating rotavirus vaccines. These focus on an outer membrane protein of the virus. This is the VP4, which is the spike of the rotavirus, and VP8 is one part of VP4. This is the variable portion of the spike protein. We are developing currently, working with PATH, a trivalent vaccine candidate, which is based on VP8 subunits. Initial studies with this have been done in South Africa. Phase 1 and phase 2 were very promising. We were hoping to go into phase 3 studies in Africa and India, and unfortunately with the COVID-19 outbreak, it seems like we might have to repeat the entire phase 3 studies all over again. So even though we had a success story, there is still a long way to go, even for rotavirus vaccines. Moving from rotavirus to Ebola, if we look at Ebola, we had a virus that was discovered in 1976 that caused repeated small outbreaks over the next 30 years, and then in 2014 caused a very large outbreak in West Africa when it came to urban centers. The World Bank estimated that Ebola cost the world $4 billion in direct costs and about $54 billion in total costs. For SARS-CoV-2, just for comparison, it's been estimated that the optimistic take on how much the world economy will lose is $3.3 trillion. The upper estimate is over $70 trillion, and the consensus is about 27 trillion, which equates to 5.3% of global GDP over 5 years. So this is just to tell you how much of an impact outbreaks can have. Moving back to why vaccine development for outbreak pathogens is very difficult is if you look at the stages of evaluation of the vaccine and you look at the cost of those vaccines, the early stages are not very expensive, but as soon as you get into clinical testing, costs really escalate, and it can cost $500 million to develop a vaccine. The probability of success goes up through all of these stages, but as many situations, vaccines tend to be developed when you know that there is a certain market. A certain market is when a vaccine is going to be used for routine immunization and not for outbreaks, where the timing and the size of what is required is unpredictable. So because of the lack of high return on investment, most multinational companies do not usually invest in making vaccines for outbreaks. And this was the situation for Ebola. The virus was identified in 1976. Jack Rose described a reverse genetic system for VSV in 1996. A replication incompetent VSV vector was developed to study Ebola the next year. And in 1999, Feldman developed a replication competent VSV vector with an Ebola glycoprotein at Canada's National Microbiology Laboratory. 2 years later, they started work on an Ebola vaccine, showed that it was effective in animals in 2005. They licensed this to a company called Bioprotection Systems in the US in 2007, but continued their own work to partner with IDT in Germany for CGMP manufacture of Ebola vaccines. In 2015, they had enough to give the to give to WHO for pilot studies of the vaccine. In 2014, Merck bought the license from Bioprotection Systems, and we now have a vaccine that is known as the Merck vaccine. This vaccine was evaluated in the Ebola outbreak in West Africa in 3 countries, Guinea, Sierra Leone, and so what happened there was the outbreak really started in 2014, but by the time the decision was made to evaluate the vaccine, it had already taken 6 months. The discussion started, but vaccination was initiated only in March of 2015. If we look at how long it took for us to get from making the decision to use the vaccine to actually using the vaccine, even though the vaccine was ready in Canada, it was a period of approximately 9 months. Within a few months of having made the decision to use the ring vaccination strategy and starting with immediate and delayed vaccination arms, within a few months, the data were available that showed that if you did immediate vaccination, you had no cases that were identified in contacts. Whereas if you did delayed vaccination, you did see cases. So the vaccine was in this study 100% efficacious, but by the time the study was actually being done, as you can see from here, there were practically no cases left. The outbreak had come and almost gone. Nonetheless, this resulted in the first Ebola vaccine, an RBSV vaccine, which was approved by EMA in 2019. It was also subsequently approved by FDA in the US. And this year, we have had the second Ebola vaccine, which is based on an adenovirus vector which has just been approved in 2020. Now, if we look at challenges related to the development of new viral vaccines, there are many, but both challenges and solutions can be seen when you have vaccines that you need to develop for epidemics and pandemics. So if we look at what was happening around the world during and post-Ebola, WHO and others were calling for global action. We know outbreaks, epidemics, and pandemics are almost predictable, but they have increased over the past 30 years because of increasing trade, travel, population density, migration, deforestation, and greater interactions between animals and humans. There is, in fact, an alphabet from AIDS to Zika of zoonotic infections that have spread around the world. So in response to Ebola, there was a new initiative for preparedness. This was called the Coalition for Epidemic Preparedness Innovations. The idea was to develop vaccines that could be used during outbreaks and to make this a mechanism where what had happened in Ebola, the delayed deployment and testing of vaccines, would not happen again. So the idea of CEPI was to accelerate the development of vaccines against emerging infectious diseases and enable access to these vaccines for affected populations during outbreaks, essentially to make new vaccines for a safer world. Now, CEPI's strategic portfolio was initially Lassa, MERS, and Nipah. And then we added Rift Valley fever and chikungunya. But in addition to disease-specific targets, there was also the idea of disease X, of developing platform technologies which could improve the speed of vaccine development against unknown pathogens. Because projects had already been funded for all of the target diseases as well as for platform technologies, within 2 weeks of the sequence of SARS coronavirus 2 being released, CEPI was able to fund 3 projects for SARS-CoV-2 vaccines. CEPI's COVID-19 vaccine platform currently consists of 9 vaccines, 8 of which are currently in clinical trials, and they are over a range of platforms from DNA to mRNA to viral vectors and proteins. If we look at the platform technologies that are available for SARS-CoV-2, every technology that we have is in use. So if we look at inactivated vaccines, we have a number of candidates where the inactivation methods are based on viral vaccines that have previously been developed for a range of different diseases. Hepatitis E virus is an example. There are also vaccines for Zika and other viruses that are based on inactivated viruses. If we look at live attenuated vaccines, I had mentioned previously that one of the new technologies that is being used is codon de-optimization. And this is an approach that is being followed by multiple vaccine companies, including a couple of partnerships that Indian companies have for this approach. These vaccines have not yet entered into human clinical trials. If we look at protein subunit vaccines, these can be virus-like particles or protein by themselves. And for SARS-CoV-2, mostly what is being used is spike or receptor binding domains, and there are a couple of Indian companies engaged in that. Most subunit vaccines, unless they are made as nanoparticles and therefore self-adjuvanting, they will need other adjuvants as well. In India, one of the most interesting and exciting efforts is something that Raghavan Vardarajan is doing with his company Minvax, where he's focusing on receptor binding domain of spike protein, and he is using the approaches of structural vaccinology, which he has used previously for HIV and for next generation influenza vaccines, but where he is going beyond what most other vaccine companies that are focusing on RBD antigens, is that he's trying to make a vaccine that can be used in a range of different settings. So the focus has really been on making a thermotolerant protein so that the cold chain requirements that most vaccines have will not be necessary for the vaccine that he produces. If we look at DNA vaccines, and I apologize for the slide moving a bit, we have Zydus Cadila looking at a skin patch to deliver the DNA vaccine. And Inovio was using its own device, which is called Selectra, to deliver the vaccine, but because of issues with the device, Inovio has not yet moved to phase 3 studies. If we look at mRNA vaccines, Moderna and Pfizer BioNTech are in phase 3 trials, and it's expected that these two companies will be among the first ones to have a readout from phase 3 studies. Genova in India is working on an mRNA vaccine, and one of their greatest concerns has been the same issue of stability that is seen with many other RNA vaccines. And they are looking to see how to work around that. An RNA vaccine that needs to be delivered at minus 70 degrees is not really a public health solution for the world. If we look at viral vectored vaccines, you can see that there are a number of candidates. But as I pointed out earlier, there are only 2 viral vectored vaccines that have been licensed so far. And both of those are for Ebola, using the VSV vector for the Merck vaccine and adeno vaccines for Janssen & Janssen. Janssen & Janssen's Ebola vaccine uses Ad26, which they are also using for SARS-CoV-2, but it also has a boost with an MVA second dose. And with the adeno vaccines, the concerns about prior human exposure to adenoviruses is something that people are really worried about. The University of Oxford used a chimp adenovirus and has shown that you can get boosting with a second dose of the same vaccine. And I thought here I would show you a little data from the University of Oxford vaccine and use that to talk a little bit about vaccine safety. So the data from the phase 1-2 study, the large study that was done in the UK, has been published in August in the Lancet. This was a single-blind randomized controlled trial that used the meningococcal conjugate vaccine as the control. They vaccinated healthy adults that had no history of SARS-CoV-2 infection or symptoms to receive either ChAdOx1 and COVID-19 or to receive the meningococcal vaccine. What they showed in this study was the presence of binding antibodies where they demonstrated that if you got the vaccine, you made good antibodies, and that these antibodies were comparable to the binding antibodies that were seen in people who had recovered from SARS-CoV-2 infection. They also demonstrated that if you did a prime boost strategy, you could boost the immune response to ChAdOx1 as measured by anti-spike IgG responses. They also demonstrated that neutralizing antibodies were made in the majority of participants, and that these antibodies were comparable to what was seen in convalescent plasma samples from recovered patients. They went beyond that to look at specific T-cell responses and showed that these peaked on day 14. Now, it's very unusual for viral vaccines to start with measuring cell-mediated immunity. But there is a reason why that was being done. And that's because very early on, it was recognized that there were potential risks that were associated with developing certain platforms as vaccines for COVID-19. Previously, there have been negative effects seen with vaccines that are either flavivirus vaccines or vaccines that are inactivated, and particularly for inactivated coronavirus vaccines evaluated in animals. And these mechanisms are believed to be antibody-dependent enhancement, which is antibody-mediated, or vaccine-associated enhanced respiratory disease, as seen for the respiratory syncytial virus, which is largely T-cell mediated. So in the guidance that the FDA and other regulators have put out for monitoring the development of SARS-CoV-2 vaccine, it's been emphasized that you need to measure not only the presence of high-quality neutralizing antibodies, but also ensure that the response to these vaccines does not have a Th2 bias. Now, where did we get this understanding from? We got it from structural vaccinology studies that were based, as I had mentioned previously, on work that had been done for HIV and then more recently for other respiratory viruses. This is an example of work that is done at the Vaccine Research Center at the NIH on respiratory syncytial virus, where they demonstrated that the VAERD that was seen, the antibody-mediated component of that was because there were weakly neutralizing antibodies that were made in natural RSV infection to the fusion protein of RSV. It was important, therefore, if you wanted to make a potent neutralizing antibody response, to have a stable pre-fusion F protein. This would allow for designing a better antigen where you could not only induce a good immune response, but also induce an immune response that was sustained over a long period of time. So therefore, structural vaccinology is one of the techniques that has allowed us to better understand safety and the making of effective antigens. This is also being used now to think about next generation influenza vaccines. If you look at chart A on the top, that shows you that with the current approaches that we have to influenza, the performance of vaccines tends to be quite variable. What we try to do every year is predict which vaccine is going to be which influenza strains are going to be circulating some months into the future, and then make largely egg-based vaccines on that. We also have now inactivated vaccines live attenuated vaccines and protein vaccines that are made every year in response to the predicted circulating strains of influenza. But this is really not sufficient. When you have strain-specific vaccines, then the amount of protection that you can get in a particular year for influenza can be variable. So if we look at C at the bottom, if we think about a true universal vaccine, what we are looking for ultimately is to have a vaccine where you can have a single product that will protect against all strains of influenza and give protection for a prolonged period of time. We are very far from that at the moment, but there are ongoing efforts around the world, and most of them focus on clues that we have from structural vaccinology. So from focusing mostly on the head, which is where the receptor binding sites are, can we think about using the stem and developing vaccines there? The ability to make nanoparticles by stabilizing stem hemagglutinins is also something that is in progress and has promise. So as we look towards the future, there are going to be a number of approaches that are taken to making better next generation influenza vaccines. So if we were really to summarize, I think what has happened with SARS-CoV-2 is that we have advanced the science of vaccinology over 10 years in the less than 10 months that we've had with this virus. Vaccines are important, obviously, for SARS-CoV-2. They are not going to be the perfect solution, but they are an incredibly important tool of public health. If we look at vaccines and what they could do potentially just for India, we can think about protecting people against a range of pathogens, respiratory, enteric, others. And that will mean that even without outbreaks, we will be protecting our populations against unexpected expenditures that drive people into poverty. Obviously, this is the most worthwhile of goals, and I hope that we will take the learnings that we have from this encounter well into other diseases that are caused by viruses and then by bacteria as well. So thank you very much for your attention, and I'd be happy to take questions.